This is Syed Suhaib Ahmed. I am going to present the topic on role of active targeting agents in the treatment of colon cancer. So, table of contents: introduction, definition, causes, treatment modalities, role of nanoparticles, strategies of active targeting agents in the treatment of colon cancer, challenges and conclusion, and specific learning outcomes. At the end of lecture, students will learn about how colon cancer will spread and what is the etiology, and how colon cancer will spread and what are the different stages. What is the role of nanoparticles in the treatment of colon cancer? To understand the strategies for targeting the colon cancer through active targeting. And next is future challenges and conclusion drawn from preclinical studies. So, introduction. Colon cancer develops as a result of the change in normal colonic epithelium to adenomatous polyps. So, it changes from stage 1, stage 2, stage 3 to stage 4. There are different stages. So, it will progress from stage 1 to stage 4. So, what are the different causes? Genetic alterations which are described by such that a change in the development of cancer which has been increasing in the recent years, such that different changes in the genetic alterations will make that to develop the cancer, and progressively it will develop to stage 2, stage 3 and stage 4. So, as in 2018, 1.8 million new colorectal cancer cases were raised, and among them 8,81,000 deaths were occurred. So, among them, which accounted for the nearly about 10% of the cancer cases worldwide. So, among them they still cases of colon cancer may rise to 2.5 million in 2035. So, what are the different causes? Obesity, crimson meat, smoking and excessive drinking are the genetic risk factors. On the other hand, which are lifestyle changes such as diet, genetic mutations, they play important role in the development of disease. Surgical resection and post-operative adjuvant chemotherapy are the most effective treatments in the treatment of this colon cancer. So, what are the different stages for the treatment? Surgery and chemotherapy are the most effective treatments used in the first line, second line and third line therapy. So, these treatments can successfully remove the cancer from micrometastasis. They can remove from the implant by using surgery method. They reduce the colon cancer rate by spreading to different parts of the body. However, despite these emergence of numerous screening programs to reduce the colorectal cancer incidence, nearly a quarter of colorectal cancers are diagnosed at an advanced state with metastasis. So, certain cases may not be reported in stage 1 and 2. So, they may be progressed to stage 4. So, among them 20% of the cases which develops to metachronous metaphases, which result in difficulties in curative surgical control and subsequent tumor related deaths. Disappointingly, traditional therapies in this stage have certain side effects and limitations. For chemotherapy, due to poor physical and chemical properties, low viability and poor tissue selectivity of chemotherapeutic drugs, patient may suffer from serious side effects. So, in the end, this targeting potential will pave the way for the development of a successful method for the management of disease and better patient care. So, for several decades, research has focused on developing new approaches and techniques for studying cancer, particularly in the area of detection and early cancer therapy to reduce the mortality. It is encouraging that with the development of nanotechnology, there are many nanodrug delivery systems targeting colorectal cancer with high efficiency anti-cancer such as lipid nanoparticles, polymer nanoparticles and liposomes, which can realize that delivery of chemotherapeutic drugs, genes and other vaccines. So, what is active targeting of nanoparticles? Ligand modified nanoparticles can actively accumulate in the tumor through a ligand receptor binding mechanism, thus delivering the drug to the target. These nanoparticles are called active targeting nanoparticles. So, cancer cells which overexpress these receptors secrete some biomolecules that can promote cell proliferation of cancer and surrounding tissues through paracrine or autocrine pathways. There are two main methods to combine ligands with nanoparticles. One is to modify them chemically during the synthesis of nanoparticles, and other is to bind ligands with polymer before the synthesis of nanoparticles. Ligand modified nanoparticles can accumulate in the tumor through passive targeting and then enter tumor cells through active targeting, resulting in a more selective and enhanced therapeutic effect. So, this is an overview where active targeting can be occur, where we can develop pH dependent polymers and ligand modified polymers to the by surface decorating to the nanoparticles, thereby when they are released into the body, so they should be protected from the GI tract. So, the different pH from GI, stomach and small intestine, large intestine varies. So, they should be protected such that until they reach the colon tissue, colonic region, they should be protected well. So unless if it is released in stomach or small intestine, no activity will be shown. So as tumor cells have leaky blood vasculatures, more amount of drug can be removed outside. So in order to prolong that, such that nanocarriers are deployed such that they are in tiny size, such that they penetrate to those blood vessels and they go and bind to the particular receptors, such that on particular binding they will show its activity. So what are the different types of receptors for targeting colon cancer? Folate receptors. Folate receptors is a cell membrane anchored protein with high affinity for binding and transportation of folic acid. It has been reported that folate receptors is consistently overexpressed on the surface of colorectal carcinoma cells and poorly expressed in normal mucus. So aptamer binding. As we know, S1411 is a well-known aptamer which specifically binds to nucleolin and has been tested in phase 2 trials patients with metastatic renal carcinoma. So researchers have conjugated the AS1411 DNA aptamer to various nanocarriers such as mesoporous silica nanoparticles and super paramagnetic iron oxide nanoparticles to mediate the selective drug delivery. Nucleolin. Nucleolin is considered as a remarkable target as it predominantly localized in the nuclei of normal cells, while it is selectively translocated to the cell surface of numerous cancer cells such as CRC cells. So this location is related to colorectal carcinoma, tumorigenesis, metastasis and angiogenesis. Because of its selective expression, targeting cell surface nucleolin in tumor cells appears to be an interesting way for anti-cancer drug delivery. And next is hyaluronic acid. Hyaluronic acid will express CD44 receptors on the cell surface. It is a transmembranous glycoprotein that exists as an adhesion molecule on the cell surface and takes part in intercellular and cytomatrix interactions, as well as cell adhesion and migration. The overexpression of CD44 in colorectal cancer is closely related to tumor metastasis. Therefore, it is also a biomarker for colorectal cancer as well as target. And nanoparticles which are based on stimuli responses. Stimuli responses it depends on mainly pH, temperature. Some nanoparticles which can achieve precisely target therapy for colorectal cancer patients under the condition of stimulation such as external magnetic field, temperature, reactive oxygen species and near infrared spectroscopy. In research, the complex of biological super paramagnetic chitosan based nanocomposite was prepared to deliver SN38 drug by combining with hydrophilic polymeric prodrug poly L-glutamic acid SN38, which showed obviously enhanced accumulation in colorectal cancer and more easily can be internalized by the assistance of magnetic field. pH dependent nanoplatforms. So among various methods, the most focused approach is to develop pH dependent. So depending on the pH of different stomach, small intestine and large intestine, these pH are designed such that they release the drug in the particular pH. The gastrointestinal tract is structurally divided into gastro, small bowels, intestine and crasum. So in addition to this difference in the physiological function, the pH of each part is also different. So pH of stomach is approximately 1 to 3 and for small intestine it increases from 5.5 to 6.8 and which is close to neutral environment, and the pH of colon is 6 to 8. So inspired by these enteric coating tablets, the researchers began to develop a nano drug delivery system containing enteric coating materials to make drug released in the colon. At low pH values, these nanoparticles are intact, but at high pH values, due to the dissolution of coating materials, the nanoparticles will swell and adhere to colon to release the drug on a specific region. So it is a different types of active targeting process where our receptors will be binding. So in pH receptors we will be using 2-polyvinyl pyrrolidine, polyacrylic acid, zinc oxide nanoparticles, chitosan based nanoparticles, beta cyclotrypsin based nanoparticles, cyclotrypsin, and in redox responsive we will be using glutathione disulfide bonds, and enzyme responsive we will be using gelatin, bilirubin, polybeta amino ester, polyglutamic acid, and in ATP response we will be using ATP aptamers. So where there are various thermoresponsive polymers also such as poly and isopropyl acrylamide, duplex DNA, and light sensitive we will be using azobenzene gold nanorods. For ultrasound responsive we will be using tetrahydromethacrylate, polydimethylhexane. For electro responsive we will be using chitosan hydrogels. So these are the various types depending on various stimuli, light stimuli, and what are the types to be focused based on these receptors. We will be using different pH based receptors. So, what are the challenges in nano drug delivery system? So, considering the high efficacy of nanoparticles in animal models of colorectal cancer, the clinical application of nanoparticles is promising. However, there are still some shortcomings of nano drug delivery systems. The biggest challenge is how to produce nano preparations on a large scale and to evaluate the preclinical safety and effectiveness. Additionally, in clinical trials, it is found that nano platforms tend to reduce the toxicity of drug rather than improve efficiency. According to the analysis of many scholars, most nanoparticles accumulate a tumor based on EPR effect. So, even actively target nano preparations, but EPR effect is more consistent among animals. So, while there are differences in EPR for colorectal cancer patients, which will affect the efficacy of nano preparations. Therefore, personalized therapy is needed, that is, nanoparticles are used in patients with strong EPR effect to achieve better efficacy, or to use preparations that can accumulate in the tumor independent of EPR effect, such as temperature response based hydrogel for local treatment of colorectal cancer. Conclusion: in the treatment of colorectal cancer, colon targeted nano drug delivery systems can change the distribution and release of the drug in humans by accumulating in colorectal cancer, which achieves a better therapeutic efficacy and reduces side effects in comparison with traditional treatment methods. Therefore, designing nanoparticles with high targeting ability has greater significance for colorectal cancer. And these are some of the references used for the study. Thank you.